of Gene and Cell Therapy. My name is Hildegard Bühning. I'm Professor of Infection Biology of the Gene Transfer at Hannover Medical School and the current President of the ESGCT. As you might already know, ESGCT is a non-profit organization promoting fundamental and clinical research in gene therapy, cell therapy, and vaccines. Education is an important part of our mission. We therefore launched the ESGCT eSchool in May this year. We are very grateful to all our colleagues who are all highly recognized experts in the field of gene and cell therapy, like our speaker today, for their support of our initiative. The current part of our lecture series is dedicated to gene and cell therapies of rare diseases, and today we focus on the CNS. It is my great pleasure to introduce to you our speaker of today, which is Professor Nathalie Cartier. She is Director of the Biotherapy for Neurodegenerative Disease Department in Paris, and she studied medicine. She focused her interest as a clinician as well as a researcher towards the development of gene therapy application for neurodegenerative diseases like leukodystrophies, Alzheimer, or Huntington. She is engaged in the whole pipeline from preclinical proof of concepts to clinical application. Nathalie has been president of our society between 2014 and 2016. She has an impressive publication record and has received numerous awards. Last year, Nathalie has received the Outstanding Achievement Award of the ESGCT, the highest award of our society. As said, today she will give a lecture with focus on gene therapy of CNS. Nathalie, we are looking forward to your lecture.
So I'm going to tell you today about some aspects of gene therapy for neurodegenerative disease, with a special focus on a program on Alzheimer's disease, but make some general consideration that I think can be helpful to you. Starting with the specific aspects of CNS gene therapy. It's general things, but I think it's worth reminding that. Of course, gene therapy is very different for CNS disease, whether you want to target disease that affects specific brain area, like for Huntington, Parkinson disease, or Huntington, where the target region to correct is the striatum. Or if you want to deal with large portions of the brain, like this is the case for many genetic diseases like lysosomal storage disease that affect all the brain, or disease like Alzheimer. And of course, the brain is a complex organ, so you have to understand the connections and the relationships between cells, and also what cell population you want to target. You want to target all neurons or specific subpopulation of cells? Do you want to target glial cells? And this is, of course, makes a big difference in terms of the strategy. And the last thing, of course, is the blood-brain barrier, because the blood-brain barrier protects, of course, the brain for many lesions, and particularly from the immunological response. But of course, it impairs the delivery of therapeutic product, and the viral vectors are mostly not able to cross the blood-brain barrier so far. So the two vectors that we are using for gene therapy, as you know, are lentivector for a few indications, but mostly adeno-associated vectors. And this is the one I will tell about today in this lecture. There are two ways to do gene therapy into the brain. One is ex vivo gene therapy, where you can take the cells out of the patients, particularly hematopoietic stem cells, and correct the cells with a vector outside of the body. And for this is mainly lentivector, because they can integrate into the cells and so have a correction for lifetime in dividing cells. Or you can do in vivo gene therapy, bringing the viral vector directly into the body, either by brain injection or CNS delivery, or by IV delivery, which is of the final goal that we all to achieve. And this is the part that I will discuss today with you. So for that, the AAV vectors are the most important vector. This vector is an interesting one because It is a small vector. It's very well tolerated in human beings. We're all affected with AAV, but with no harmful consequences. And there's many serotypes of this vector that allow very different targeting into different cell types, and particularly into the brain, as you see here. One is missing here, which is AAV-RH10, which we use a lot currently for CNS indication, and I will tell you more about it in a minute. Another important thing to mention is, so here you have an example, excuse me, of the different serotypes expression where you do exactly the same injection and you see the different pattern of expression you can get in different regions of the brain based on serotype. Also, what you can do to specifically deliver your therapeutic gene in one cell type or the other is to change promoters. And of course, by using different specific cell type promoter, you can have expression in neurons or in glial cell. This is just an example, comparing with the same therapeutic gene, one housekeeping promoter PGK, and the promoter GFE2 that leads to expression specifically in astrocyte. And you see with the same injection of the same titer, a very different pattern of expression with neurons mostly for the PGK and astrocyte exclusively for the GFE2 promoter. So now going to in vivo delivery, the two main strategies that have been used so far is either intraparenchymal delivery or intra-CSF delivery. And this is what I will tell you most about it. This started with this, one of the first applications in human patients was for Canavan disease. And you see this is from 2002. And this is really a pioneering work performed at that time. And the very interesting thing is, if you look at this paper published 10 years afterwards, and that show in this clinical trial that they have been a lot of safety data collected. Here you see safety data collected, a minimum 5 years follow-up, and showing a good tolerance and therapeutic efficacy with a decrease of a biomarker, which is a NAA into the brain, and the slower of the brain atrophy. So the first really important positive and encouraging result into direct brain gene therapy. So I put that, of course, this is not for you to read right now. This is a paper in Press in Human Gene Therapy. We have made this recent review to see what has been done so far in clinical trials for intraparenchymal injection, as you see here, with different vectors, mostly AAV, as you see. One is lenti here, but most of the others are AAV, and with different kind of application. But as we will see also in this paper, most are still lysosomal disease, like here Batten, Canavan, but or MPS III, but also a lot of applications for Parkinson's disease. And again, Parkinson is a very strong indication because it's a local disease of the striatum, and more recently, Huntington disease. There have been also some intraparenchymal administration here in this. So, excuse me, this was all the safety preliminary steps that were performed, and these are for the intra-CSF and intravenous administration into human patients, where you see that there's much less clinical trials ongoing, but still an interesting number of IV delivery. But what you can see from this slide, importantly, is the dose that are used compared to the last slide, which was intraparenchymal. I'm sorry, this is a mistake on my slide. It's not intraparenchymal, it's CSF or IV. And you see that the main difference, of course, is the high dose that are needed to go into CSF or into intravenous delivery, where you need 1 or 2 or even 3 log more vector to be able to have a therapeutic efficacy. So one important point I want to mention now, and this is what I will describe in the next slide, is that gene therapy actually is not restricted to genetic disease. Gene therapy for genetic disease is quite straightforward, of course. This is based on gene replacement to correct a loss of function of a missing protein, but it can be also gene silencing to stop the expression of a protein that has toxic function, so correct dominant gain of toxic function, or of course this can be more recently gene editing strategies. But this can be also indirect gene therapy to improve or to restore deficient cellular pathway. This can rely of bringing back growth factor or correcting neurotransmission pathway, or more broadly, correcting the deficits into different metabolic pathways, and particularly energy pathway that affect the brain very importantly. And the comment that I want to make on that is, again, that this can be applied to all diseases, genetic and multifactorial diseases. But because this is less straightforward, that one gene missing, you put back the gene, this needs a very strong validation of the therapeutic target when you do the preclinical proof of concept of your strategy. The final comments on that, and this goes very well together with the last slide, is that for rare disease, and particularly for very rare disease, we need to find treatment that can be beneficial to several diseases. Because for several very rare disease, it will be very difficult to implement gene therapy strategy up to clinical application in patient because of the cost, because of the difficulty to set up all these clinical strategies. But this could be achieved if we can identify and target common physiological pathway that we can target to treat these very severe pathologies. And of course, this can be, if we do that to very rare disease, this can be also applied for complex multifactorial disease. And this is what I will describe now in this second part of my talk. I will take the example of what we are currently doing in my lab, which is evaluating one specific target, which is the brain cholesterol metabolism as a therapeutic target for neurodegenerative disease. Why did we focus on this particular metabolic pathway? It's because it has been clearly shown now for many years that cholesterol is strongly involved into neurodegenerative disease. And it has been well shown, particularly in Alzheimer disease, where you will see in a minute, but the link of lipids and cholesterol metabolism is really important, but also in other diseases like Parkinson or with Huntington disease. So we've decided to focus on this pathway and on cholesterol into the brain. Cholesterol into the brain is an extremely important component. Our brain is 2% of our body, and it contains 25% of cholesterol. It's mainly into the myelin sheaths, as you see here, and the 1/3 is in the neuronal membranes, in the synapses. And this is really the important part I want to tell you about today, because this has to go with all the signaling and the synaptic functions of neurons. And the composition of cholesterol into these synapses and to the membrane is critical to ensure the signaling, to ensure the synthesis of some proteins, to ensure the right fluidity of membranes that makes the neuron functional. And as you know, when neurons are dysfunctional, when synaptic function is not working any longer, the neurons finally dies. So this is the cholesterol we are interested in. So we've been interested, we started with that with Alzheimer's disease. Why? Because again, it was really straightforward. Many things were known about this link between cholesterol and Alzheimer. The most important one was the role of ApoE. You know that ApoE is the main risk factor in Alzheimer's disease, and ApoE is the main transporter of cholesterol into the brain. And more recently, it has been shown that not only ApoE is implicated in Alzheimer's disease, but also in the GWAS analyses that have identified some other risk factor for Alzheimer. Several genes have to do with the cholesterol and with lipid metabolism in the brain. Another important thing is that cholesterol is increased in the brain of Alzheimer's patients, and that there is a strong link between the composition of membrane in cholesterol and to the production of A-beta. And what is important for the following elements of our strategy is that the product of cholesterol efflux out of the brain, which is called the 24-hydroxycholesterol, is decreased in the plasma of AD patient, meaning that the metabolism is impaired in the brain. And the gene you will see in a minute responsible for this metabolism has shown some gene polymorphism associated with Alzheimer's disease. So this is the big picture. This is why we were interested in cholesterol metabolism for Alzheimer and in the role of this specific enzyme. So this enzyme is the hydroxylase of cholesterol, the 24-hydroxylase of cholesterol. This is the whole pathway of cholesterol. I don't go, of course, into detail. This is not the point. But just to show you that this is absolutely necessary for cholesterol to cross the blood-brain barrier and be eliminated in the periphery, because cholesterol cannot cross the blood-brain barrier. So it is produced in the brain, and it is eliminated in the form of 24-hydroxycholesterol. So this enzyme is really the key enzyme of cholesterol metabolism in the brain, and it regulates the whole pathway. It activates the whole pathway. And you see on this slide, this short summary that tells you that all the compounds on this pathway have all critical roles in the function of neurons. And particularly look at that, synaptic transmission, memory, trophic role, autophagy. So you see that all these compounds are directly implicated. And so this explains to you why this enzyme is so important. And again, it has been associated with Alzheimer's disease. So what we've shown first to try to evaluate if this enzyme could be a therapeutic target for Alzheimer's disease is there a link between this enzyme directly and the pathology in patients? So we were lucky to have samples from patients who died from Alzheimer's disease. And in this patient, we could show that indeed in the brain, the enzyme is decreased in the neurons. You see here the expression using histochemistry in disease compared to normal individual. And you see here the level of the enzyme, which is decreased. And you see that it is also decreased in mouse models of Alzheimer's disease. You have two examples here. One model of amyloid pathology, which is called the APPPS1 knock-in, decreased enzyme, and in the tau mice, which is a model of the other part of the disease, which is the tauopathy, again showing the decrease of the CYP46 enzyme. So this was a strong argument to think that maybe if we could replace this missing enzyme into the brain, this would be beneficial to the patients. I recall there is polymorphism associated with Alzheimer's disease. So we developed a strategy to evaluate this option and to show two different things. One was, what are the consequences of the inhibition of this enzyme in a normal brain? Is it leading to harmful effects or not? And second, of course, can we correct some of the pathology of Alzheimer by overexpressing the enzyme? So I don't go into details. I prefer to tell you the global strategy. The details are published and you can easily go to the papers. So this is the part on the inhibition. And you see that here we published, this is a work of Fatia Gelti, who has a PhD in my lab, and she demonstrated very elegantly that by inhibiting the enzyme, we have very, very powerful effects, and leads to the accumulation of the amyloid pathology. And this was also published by Farah Shali, who is now postdoc in my lab and was a PhD student at that time, showing also that by inhibiting this cholesterol degradation into the brain, we have very severe consequences, particularly on epileptic activity on the mouse hippocampus. Briefly, this is what we have shown. Injection here in the normal brain of the vector with a shRNA against CYP46 here in the hippocampus that you see here. This, of course, decreased the activity of the enzyme, decreased production of 25-hydroxycholesterol and content here, and decreased expression of the enzyme, increased cholesterol. And by doing that, interestingly, what we see is very severe neuronal death in animals, hippocampal atrophy exactly as we see in Alzheimer's disease, and memory defect exactly as we see also in Alzheimer's disease, with an increased production of A-beta peptides. So really, this mimics to some extent what happened in Alzheimer's disease. So here you see the accumulation of the amyloid product. And also what we discovered is that when we purified lipid rafts, we could show the increase of cholesterol and increase of APP in specifically these lipid rafts that alter profoundly the capacity of the amyloid destruction of this in this mice, and also the increase of the tauopathy. So second part was the overexpression in different animal models of Alzheimer's disease. Here by using an AAV vector carrying the CYP46 cDNA, and to do so and to confirm the relevance of the strategy, we used several models, 3 of amyloid pathology and 1 of the tauopathy. And again, I emphasize that this part is extremely important when you deal with a disease where you test an indirect therapeutic target, and you have to demonstrate that is relevant and this is well tolerated. So this was published in 2 papers by Héloïse Udry, who is now associated professor at Harvard. She was my PhD student at that time, and Marianne Burlot, who worked on the tau model. Héloïse worked on the amyloid model. And interestingly, we saw exactly the same correction in these 2 models, the 2 components of the disease, where we could see that not only we decrease the accumulation of amyloid, toxic amyloid in mice, with here you see the decrease of the plaques, and here the soluble content, but also we correct the memory defects by using different memory tests. And importantly, we've been in more detail, in more depth, to show that there is some physiological consequences, and we measured synaptic dysfunction. We measured long-term potentiation, particularly here, and showed that this was improved. We showed the restoration of the spines. Here you see the spines on the normal mice, here on a diseased mice, and here you see the correction and also the dendrites, the arborization of the dendrites that is restored. This is work performed by Sandro Alves and Emilie Rey with me more recently. So really, we have now the big picture of how this correction is important on the different on the 2 different pathways, the tau and the amyloid part. So based on that, we decided this would be interesting for develop a therapeutic strategy, of course, in patients. So why gene therapy, which is not the simplest way to go, because there's no small molecule to do what we've shown with CYP46. It cannot be delivered directly, and we need to express of course, the gene into the brain, which is the target region that is affected by the disease. And because, of course, gene therapy would allow us to have one-time treatment to deliver the treatment and be efficient for hopefully lifetime. The problem, of course, we have is that, as you've seen on the first slide, most of the attempts today are based on CNS direct injection, because IV delivery is limited to certain indication and particularly efficient in very young patients, which is not the case, of course, for Alzheimer patients. So what we've done was first to try to evaluate what would be the consequence of overexpressing the enzyme into a large brain. And for that, we used a macaque, Macacus fascicularis, to show that we could go from this mouse size to patients by having a strong biodistribution of the therapeutic protein in the brain. I want to emphasize that this is not a secreted protein. So this is not like for lysosomal storage disease, where when you deliver a gene with an AAV vector in a cell, you know that this enzyme can be secreted out of the cell and recaptured from other cells from the environment to increase the therapeutic benefit. This is not the case for intracellular protein. So you really have to have a strong biodistribution to be able to hope for a therapeutic effect. So we did this administration in these primates in the hippocampus that you see here and also in the cortex. And what we saw was first very importantly that we had no toxicity in the brain. This is the MRI 2 months after surgery. And we had a very good targeting of the hippocampus and of the cortex. And here is the cortex, and here is the hippocampus, and here the hippocampus. And this targeting was very neuron specific, as you see here, with the neuron staining and the HA, which was the tag we used on our gene. So, of course, this intraparenchymal delivery is something we are currently doing. And I showed you this slide showing Michel Zerra, who is a neurosurgeon. Michel Zerra is the head of the neurosurgery unit in Necker Hospital in Paris. And Michel Zerra has been the one doing all the clinical trial with intraparenchymal delivery in France for MPS A, MPS 3A, MPS 3B, and metachromatic leukodystrophy, and also the phase 3 clinical trial for MPS 3A recently, where Michel has injected patients in France, in UK, and in US. And you see the device that Michel is using, which is called neuronavigation, which is extremely precise and allows him to go exactly on the point that where he wants to deliver the therapeutic gene with a very thin catheter that you cannot see precisely on the screen. But so good efficacy and no side effects so far in what Michel has performed in patients. But what you see here for Alzheimer's disease is that the complicated situation for Alzheimer's disease is that you have a very strong diffusion of the disease. This is the evolution with time, coming from here in the hippocampus to the whole cortex here. So of course, this is something that would be difficult to achieve. The diffusion would be difficult to achieve by AAV administration and difficult to achieve in old patients with a very fragile situation. So this is something for which we have worked a lot, thinking of alternative delivery system. And of course, the one that would be the best would be IV delivery. And we have done a lot of work to evaluate such strategy using a serotype that can cross the blood-brain barrier. And we have shown with this phenotype in mouse model that we can indeed correct the disease. Now we know that this phenotype is probably not efficient enough to go to patients, and we have to work on other capsids more able to cross the blood-brain barrier and be efficient in human patients. So this is ongoing. So, but I want to in this last third part to give you some insights of again the important thing is we have identified a pathway that is a common pathway for neurodegenerative disease and could be efficient for several diseases. And because of that, we are now focusing on another disease called Huntington disease for two reasons. Why is because of the strong link of cholesterol, I will show you in a second. And second, because this disease affect specifically one region of the brain, which is the striatum. You see here the striatum normal, and you see here in the patient the destruction, the atrophy of this region caused by the disease. And so because of the role of cholesterol in this Huntington disease, we have worked a lot during the past years with the group of Sandrine Bétu and Jocelyne Caboche in Paris to demonstrate that this enzyme is a very strong target for Huntington disease. And so this, again, are the important papers showing why we think the target is relevant. And if you look into the hallmarks of Huntington disease, which is strong dysregulation of transcription, impaired metabolism, and particularly energy metabolism, axonal transport defect, excitotoxicity, all leading to neuronal death, we know that the dysregulation of this cholesterol balance and the decrease of the CYP46 enzyme are central into this process. So I don't go again into details because this is published, but again, we have used two different models of mouse affected by Huntington phenotype to demonstrate that this enzyme is a strong, relevant target for Huntington disease. And because of these results, we are now able to move forward and propose a clinical trial for Huntington disease based on intrastriatal delivery of this CYP46 enzyme delivered with an AAV vector. And this is a trial that is sponsored by AskBio that we are preparing right now. So I finish by saying that this was importantly demonstrated us that we again have identified a common target. This is what we are doing with CYP46A1 in Alzheimer's disease, where we decrease the amyloid protein, here the amyloid plaque, but we've shown again that for Huntington, we also strongly decrease the number of aggregates and the level of soluble mutated form of Huntington, which is extremely important for Huntington disease. What you see here is exactly the same level of decrease that you see in the silencing approach that are developed by other groups, which is important. And the second important thing is that with this strategy, we do decrease the Huntington protein to the same level, but we only decrease the mutated form of the Huntington protein, and we preserve the normal form, and I will tell you why in a second. And based on these results, we were interested in looking at in other diseases based on the same kind of physiopathology, which is the accumulation of polyglutamine proteins in the Huntington protein. There are several other diseases called spinocerebellar ataxia in which you have exactly the same thing. And so we tested in this disease whether CYP46 could be a good target. And indeed, we saw exactly the same correction of the accumulation of the toxic protein. And I don't go into detail, but we have demonstrated the correction. This has been published also in 2019. So the mechanism through which this has been possible is the fact that restoring the enzyme, restoring the cholesterol metabolism in these different diseases restores one critical pathway in the cell, which is the clearance of accumulated abnormal misfolded protein by a mechanism called autophagy and proteasome. And we could demonstrate that indeed, this is known, this autophagy pathway is strongly impaired in these diseases, and that by injecting AAVC46A1, we do correct this pathway. This is the marker showing the correction, the LC3 protein here, and also the normalization of autophagosome, which maturation is strongly impaired in these different diseases diseases, we see that we do restore the maturation of this autophagosome in the cells from Huntington or SCA disease. So this is one of the important mechanisms through which we can have this beneficial effect. And if you look into details to what all these proteins do, the aggregation of abnormal protein do to neurons, that affects the clearance pathway, as I described, but also transcription, because it traps transcription factor. It affects dendridogenesis and synaptic transmission. It affects vesicular transport and endocytosis in all these diseases. And what we do by bringing back the enzyme and by correcting the metabolism, we improve all these different pathways. And this has been demonstrated in Alzheimer's disease, in SCA, spinocerebellar ataxia, and in Huntington disease. Because what we think is we can, by this strategy, correct a toxic loop that leads to degeneration in all these diseases by the accumulation of the misfolded protein aggregates and the role of on the cholesterol pathway. So this is important, and this is the end of my talk, because I go back to the spinocerebellar ataxia. Spinocerebellar ataxia, again, is a family of polyQ disease like Huntington, and some of which are extremely rare. The most frequent one is the SCA3, which is called Machado-Joseph disease. But there are others, as you see here, which are very rare. And like what we have shown, and this is the work of Sandro Alves, what we have shown is that by using two different models of SCA3, Machado-Joseph, indeed the restoration of CYP46A1 corrects the disease by restoring the Purkinje cells that are severely affected in Machado-Joseph disease, by decreasing the ataxin-3 aggregate, by improving the cerebellar volume, which is atrophied in this mouse model, and of course, by restoring to some point the behavior dysfunction of these mice. So the conclusion is, based on one pathway, you could propose one gene therapy product for several rare diseases, and particularly several very rare diseases. So I think it's, for the next step, this is important. So I finish by summarizing key things that I would like you to keep in mind for brain gene therapy. We think that gene delivery to the brain is safe. Be careful when I say it's safe. It means like if we work very hardly to identify the right target with the right dose and the right route of administration, gene delivery to the brain is safe. Long-term therapeutic expression of the therapeutic protein has been achieved in patients, and I've shown you the first. I started with the Canavan disease and these results, but now there are results in different diseases like Parkinson or even Alzheimer. The very early stage showed that the expression was more than 15 years in large animal models or in patients. Clinical efficacy has been evidenced in some of these diseases, particularly lysosomal storage disease, but also to some point in Parkinson's disease. And tools are being improved. New serotypes, AAV, more potent to be injected IV in particular. Also technical improvement to improve the efficacy of brain delivery. An example is opening the blood-brain barrier, which could be combined to IV delivery. And of course, a critical part, which is the important improvement of manufacturing of AAV vectors, because of course, we need a lot of AAV to imagine injecting vectors IV into the patients. I told you it's like 2 log difference between that and intrathecal delivery. And of course, we need to decrease the cost, so these treatments can, at the end, be beneficial to a very large number of patients, and not only to few patients in phase 1 studies. And this is really what has been developed in the very recent years. And of course, there is a problem of the immune response, which is unsolved today, on which many groups work, and with very important improvement by different strategies. It is not the aim of my talk, but you have to know that this is really addressed today and will be able probably very efficiently to inject IV vectors with no immune response. We still have some obstacles to solve. One important you should have in mind is that we need relevant models, because we're working on mouse models, which are extremely artificial models. And in many times, they are models of the very late stage of the disease. They are caricatural, and they are not focused on the key pathways that affect this degenerative disease, so this long-term pathology, for which what we need to address is the mechanism, so the early stage of the disease. We need to improve clinical trials to identify therapeutic benefit, and particularly identify the right biomarkers, the right endpoint. And this is extremely difficult for the brain, particularly because the brain is a difficult organ to target. It's protected. You cannot go and do a brain biopsy to see whether you have expression of your target gene. Also, because you have to treat a very small number of patients in phase 1 studies, and it's difficult to see an effect in a small number of patients, and you have to make clinical trials in a very short period of time if you want to be able to finance it. So it's really critical to have the right endpoints to show to some point efficacy, and of course demonstrate safety, but also demonstrate to some point some efficacy. Final conclusion is treat early, but this is true for all degenerative disease. This is particularly true for neurodegenerative disease, because when neurons are dead, you cannot restore them. And so when you have symptoms of the disease, whether it's Parkinson, Alzheimer, Huntington, you know that at this point you have lost a huge percentage of your neurons already. So you have to treat early, so make the detection early. Also, I didn't speak of glial cells, but astrocytes and microglia are evidently a very strong targets for gene therapy. And I finish by again repeating that we need to evaluate therapeutic targets for multifactorial disease based on cellular pathways mechanism. And this not only will be helpful for multifactorial disease like Alzheimer, but also for very rare disease for which is extremely difficult to develop therapeutic strategies. So this is where we are. The next step will be probably a lot more IV delivery, a lot more new technical tools to improve and be associated with IV delivery, focus on complex disease. And for that, what we need is really what is on the right side, which is the toolbox. I mean by that, new capsids, new vectors, new cassettes with new promoters, with improved sequences to improve efficacy and safety, but also improve manufacturing. And this is exactly what is happening today. And also focus a lot on understanding disease pathway, understanding physiopathology to be able to treat more efficiently a number of diseases. Okay, so I will stop here. You see on the slides my lab with all the people that have participated to all of that for so many years. So I will not mention all of them, but I wish to warmly thank each of them for what we have been able to do. And again, I wish to emphasize that because of the collaboration we have with AskBio, we will now be able to move forward to clinical trials today, which is extremely important for us as and for patients. Thank you very much.
Thank you very much, Nathalie, for this brilliant talk and for sharing all these fantastic data. And we have already received the first questions, which I'll tell you in a minute, but allow me to ask the first question. So I really like your view that we should, if possible, have one therapeutic gene for multiple diseases. And I really like also your point, how important it is to really do the fundamental research. One question which comes to my mind when you say we need to treat the patients early, as we all know, is what about biomarkers and science which allows you to really detect patients then in need?
Yeah, so biomarkers is really the very important issue, because what you want to see in brain disease is really difficult to achieve. If you want to see a clinical benefit in the short term in neurodegenerative disease, it's really something extremely difficult. If you want to have clinical trials on something like 1 year or 18 months follow-up, short follow-up. Of course, we follow the patient much longer, but if you want to see something rapidly. So we have to develop biomarkers, which are biomarkers of mechanism and biomarkers of disease stabilization. And this is linked to the pathway that are targeted. For example, here, what I've shown, we do have biomarkers that show us the cholesterol metabolism in the periphery. We can identify some compound that could be measured in the blood or in the CSF that directly comes to this, the correction of this pathway. We also have the imaging biomarker that allow you to identify correction into, for example, the spectroscopy profile of the metabolites into the brain and show a correction. And also we have some markers which are less specific, but that demonstrate that you have an effect on the disease progression. For example, we now know that there is a blood called the neurofilament light chain that is increased and show when you have an axonal degeneration. And if you are able to show a decrease of this biomarker, this is, of course, attesting efficacy of your treatment. So based on a combination of exploratory markers, imaging, biology, metabolomics, lipidomics, and different aspects of the disease, we will be able to demonstrate efficacy over safety.
Thank you. So let us come to the first question. So Kuna Das was asking, what was the serotype which you used, Nathalie?
We used AAVrh10. This serotype, we have used it before when we did the clinical trial for metachromatic leukodystrophy. This is a serotype that has been also used by Ron Crystal in Cornell, particularly for Batten disease. We have collaborated for years with the group of Ron Crystal. And this is also a serotype that has been used for MPS III. So, and for during the lysogene trial for MPS IIIA. So Sanfilippo disease. So it's a very strong phenotype for CNS.
And Shefali Trapati is having 3 questions. Starting with the first one. Could this process be done with any AV serotype?
It has been shown that several serotypes are good candidates for CNS. The best probably are AV9 and 10, RH10, but there are also a lot of applications done with AV2 and AV5. And the list is not, of course, limited. And there's a lot of groups working today on identifying new capsids that, again, are more specifically targeting the brain, give less immune response, and eventually are able to cross more efficiently the blood-brain barrier.
The second question is, in terms of clinical application, are there multiple doses required?
So the good thing with gene therapy is normally you hope to have one delivery for lifetime efficacy, particularly for the brain, where you know that cells are not dividing much, so you know that you have long-term expression, and this was confirmed in large animals like dogs and and primates, and also in patients. So normally we think of one administration. Of course, if you think, for example, of disease early in life, and you imagine that you might be willing to retreat patients because of growth and because of time, this is an important issue. And there's a lot of work done today to be able to re-administer the vector, which is, of course, a very important result, because this will help us to treat more efficiently and for lifetime patients.
And the last and third question of Shefali is concerning immune suppression. Is this required to achieve successful delivery of genes?
So this is
the brain.
This is the most difficult question that we may have. Of course, intraparenchymal delivery is supposed to be safe because it is said that the brain is immunoprivileged. So to a certain point, when you deliver the gene into the brain, you have immune tolerance. I say to a certain point because this is not obviously absolutely true. What is important is you have two things. You have the capsid that can lead to immune response, but it is in the short term because, of course, when you deliver the gene, then the capsid is out very rapidly. And you have the therapeutic gene. So there is the important thing to know. If you treat genetic disease where the child has no protein at all, then of course you can have a very strong immune response. If you think of a therapeutic gene that is endogenously expressed into the body, then you don't have a therapeutic an immune response. So the use of immune suppression is discussed on these elements. And you must know that there have been the two things done. Some groups have used immunosuppression, some did not. And it's very difficult today to know whether this immunosuppression has been an important part of tolerance or not, and of success. So, and again, because you cannot go to the brain and look locally what happens, it's very difficult to know.
The next question is from Victor Sanchez Clares, and it's which could be the effect of overexpression of CYP46A1 in the liver when you do an intravenous administration?
So I like, thank you very much for this question. Actually, it's a very good one, of course. And we did that because this was our first question. If we one day want to go IV, what would be the consequences of strong liver expression? And the interesting thing is, you have to know that this hydroxylase with an S of CYP46A1, of cholesterol, are present in all our cells, because all our cells are able to hydroxylase cholesterol. The 24 is specific of the brain, but in our cells we have 22, we have 27, we have 25. So we delivered a high dose of AAV carrying CYP in IV to see whether we would have any consequences, and we had no consequences. We see strong expression in liver cells, but we have no effects on hepatocytes, and we have no effects on peripheral cholesterol metabolism.
And also not on autophagy in the liver?
There's autophagy, of course, in the liver. And we have not looked, this was done a long time ago. So it would be interesting to see, for example, in conditions where you have impaired autophagy, what would be the consequence of overexpressing the enzyme in the liver. This has not been done.
Okay. Thanks a lot. So I don't see any further questions. Can you just give me your last slide, Nathalie?
Yes.
And I really would like to thank again you, Nathalie, for this excellent talk. and also our audience for the questions. And I would like to invite our audience to join us next week when Gloria will speak about deliver the main target in gene therapy. And bye-bye. See you next week.